So I see you say you have my permission?
Yes.
You have my permission to film. Hi, I'm Nicole Bodie. I'm a fourth-year grad student at Indiana University School of Medicine in Indianapolis, Indiana. And I'll be presenting my research on the human donor eyes under simulated microgravity. As we know, SANS is a main factor of vision changes in astronauts. And so we're interested in looking at how we can recreate elevated intracranial pressure in these astronauts so we can evaluate changes of SANS, specifically looking at oxidative stress markers, as well as looking at our therapeutic coenzyme Q10, which is an antioxidant, to try to mitigate these SANS effects. As you can see, we have cadaveric human donor eyes that we receive in the lab. For this study, we have 5 donor eyes that we have received, and we have one eye that is untreated and one eye that is treated, so we have a paired control. And we do a variety of different downstream experiments to evaluate these changes to these eyes. With our novel model, the translaminar autonomous system, which was developed by my PI, Dr. Tasneem Sharma, we're able to recreate normal interocular pressure and mildly elevated intracranial pressure, which is documented in astronauts on this spaceflight. The first thing we wanted to look at when evaluating this was to look at apoptosis markers, both pre-perfusion and post-perfusion in the eye. Pre-perfusion, we saw no differences between the right and left eye, and we saw the same whenever we treated our eye, that there was no differences pre and post-perfusion or post-perfusion in the treated and untreated eye. However, we saw that, and this was validated by qPCR, that there was increase in retinal ganglion cell marker RBPMS in our treated eye, and this we can appreciate with immunofluorescence. Next, we wanted to look at oxidative stress markers. So we looked at SOD2 specifically. Pre-perfusion, we saw that there were no differences between the right and left eye. However, post-perfusion, we saw that there was a significant decrease in our Western blot of SOD2 protein, indicating that there could be less oxidative stress happening in these treated eyes that are treated with CoQ10. However, interestingly, we saw that there are no differences at an mRNA level in oxidative stress. Next, we wanted to look at, with Western blot, at secreted extracellular matrix proteins. We saw there was a significant decrease in collagen 4. As known in SANS, there is a decrease, there's changes to the collagen remodeling at the optic nerve head. So in our conditioned media, we saw this was validated as well. But we saw there was decreases in this whenever we added our treatment CoQ10. However, we saw no changes in fibronectin with modification. Lastly, we wanted to look at what's happening on electrical activity level. So we used an electroretinogram under scotopic conditions, and we saw that at low flash intensity at 100 MCD, that there was a highly significant increase in the CoQ10 treated eyes with electrical activity. But we saw no differences, but with trending increase in the CoQ10 treated eye with a higher flash intensity. So from this, we concluded that there is increased interretinal function, decreased oxidative stress protein, and decreased secreted collagen 4. Now, one thing we know from our astronauts is that they have genetic predispositions to the 1-carbon metabolism pathway, and which causes a vitamin B12 deficient phenotype. We were able to get a vitamin B12 deficient donor in our lab, and with this donor, we saw that they would have an outlier in oxidative stress marker in their untreated eye, which was then downregulated in this with this treated eye. And we saw the reverse in the collagen 4 remodeling, where they had much more collagen 4 deposition in the optic nerve head, which we found interesting, and we're very excited to get more vitamin B12 deficient donors in our lab to evaluate these changes, since this phenotype is very similar to that of the astronauts going into space who developed SANS. Lastly, just to give a hint of what we're doing in the lab now, we went to Brookhaven National Laboratory in October, and we were able to radiate human-derived retinal organoids from Hispanic and 2 Caucasian donors in our lab, and we were able to do treated, untreated, and treated eyes at 2-hour, 48-hour, and 8-day harvest points. With this, I would like to thank you for your time. I appreciate it, and please feel free to email me with any questions. My email is nbodi@iu.edu.
Thank you.
Thank you.